Good day, says Professor Will, CBMD PhD. Today's Monday, June 28, 2021. It is 1:59 and 50 seconds a.m. Eastern Standard Time. Today's research, Dengue vaccine. And don't forget to check out my website. So let's hop to it here. Researchers discover how dengue vaccine fails to protect against disease. Date June 25, 2021. Source, University of North Carolina, Healthcare. Researchers discovered that a small subpopulation of antibodies binding to unique sites on each serotype are linked to protection. The research provides important information for vaccine developers to consider when creating a dengue vaccine, which has long eluded scientists. To help vaccine developers overcome this hurdle, researchers investigated samples from children enrolled in dengue vaccine trial to identify the specific kinds of antibody responses that correlate with protection against dengue virus disease. In doing so, the researchers discovered that a small subpopulation of antibodies binding to unique sites on each serotype are linked to protection. Researchers provide important information for vaccine developers to consider when creating a dengue vaccine, which has long eluded scientists. The 4 dengue virus serotypes are mosquito-borne flaviviruses that infect hundreds of millions of individuals each year in Southeast Asia, Western Pacific Islands, Africa, and Latin America. Though rarely deadly, the virus can cause severe illness, especially when a person who has previously infected with one serotype and recovers is then infected by a second serotype. This happens because antibodies from the first infection help the virus replicate during the second infection through a process called antibody-dependent enhancement. A dengue vaccine-induced antibody response weighted toward a single dengue virus serotype can mimic this phenomenon. Multiple vaccines have been in clinical development for years, and both show that they induce neutralizing antibodies against all 4 serotypes. Yet, research has also shown that the creation of neutralizing antibodies alone does not correlate to protection against clinical disease. The De Silva Laboratory conducted experiments to compare the properties of antibodies against wild-type dengue viruses and the properties of antibodies produced by a leading vaccine candidate, Dengvaxia, which the pharmaceutical company Sanofi Pasteur created using all 4 dengue virus serotypes in one formation. Researchers showed that wild-type infections induce neutralizing and protective antibodies that recognize a part of the virus, an epitope unique to each serotype. The vaccine, though mainly stimulated neutralizing antibodies that recognize epitopes common among all serotypes. In vaccine trials, these antibodies did not protect children from dengue. In the past, researchers have considered all dengue neutralizing antibodies to be protective in people. This appears not to be the case, according to this UNC-led research. The research results suggest that a safe and effective dengue virus vaccine needs to stimulate neutralizing antibodies, targeting unique sites on each of the 4 dengue serotypes. Researchers said not merely the neutralizing antibodies against cross-reactive epitopes common to all 4 dengue types. Good day, said Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.